Welcome everyone to this edition of the PerMedCoE webinar series, where we will feature the poster prize winners of the PerMedCoE Summer School that happened in June this year in 2023. Our presenters today are Viviam Bermúdez and Marco Fariñas from the Norwegian University of Science and Technology, and Malvina Marco from INSERM Toulouse Cancer Research Center. They will all present their research, and it has the common topics today will be computational modeling in different areas of cancer research. My name is Marta Llorell Linares, and I'm involved in the PerMedCoE project on behalf of MBLBI. I will be hosting this webinar. The webinar will be recorded and will be shared afterwards on the PerMedCoE website and on our YouTube channel. And after the presentations from the three speakers today, we will have some time for questions from the audience. So if you have any questions, you can write them down in the Q&A function of Zoom, and you can write them down during the presentations or at the end. All the materials that we will share from this webinar are licensed under a CC BY 4.0 license, except where further licensing details are provided. PerMedCoE is the HPC Exascale Center of Excellence for Personalized Medicine in Europe, and it focuses on simulation of cellular mechanistic models, which are essential to translate omics data into medical action. But the performance of the current simulation software is still insufficient to tackle certain medical problems, such as tumor evolution or patient-specific treatments. So PerMedCoE will scale up the cell simulation software to pre-exascale systems to enable the creation of models of cellular functions of medical relevance. And to achieve this goal, it's working on several objectives. So PerMed is optimizing selected cell-level simulation software to run in pre-exascale platforms. It is also developing a series of use cases that will show how we use the PerMedCoE solutions to address problems in different areas of clinical relevance, such as drug synergies for cancer treatment. PerMedCoE is also working on training biomedical professionals in the use of these PerMedCoE tools, on integrating personalized medicine communities into the European exascale ecosystem, and in building the basis for the sustainability of the PerMedCoE. Our speakers today are Viviam Bermúdez, a PhD candidate at the Norwegian University of Science and Technology, which will present towards Tailored Cancer Therapies, robust platform for drug testing in patient-specific Boolean models. Marco Fariñas, a PhD candidate at the Norwegian University of Science and Technology, who will present computational modeling of high-grade serous ovarian cancer, and Malvina Marco, a postdoctoral researcher at the Toulouse Cancer Research Center, who will present dissecting cellular communication in the tumor microenvironment through multiscale dynamical modeling. And now I'll leave the floor to Vivian.
Hello, my name is Vivian. I'm a PhD candidate, as Marta said. I'm gonna briefly talk about my project that is making a platform for drug testing in patient-specific Boolean models. And now first give you a brief introduction why we do this. We know that each oncology patient, let me see my pointer, each oncology patient can develop the disease in a unique way. And then functional precision medicine offers crucial opportunity in oncology, allowing treatment design and testing in patient companion models. However, with all the possible treatments and combinations that can be done, the question is, how can we predict optimal drug effects and synergistic drug combination in each patient? So at our research group, we want to accomplish this by developing computer models to test and select optimal therapies that can be tested in the lab. To do this, we have chosen Boolean models to represent the cancer cell dynamics. Then, to make the models unique, we adapt them with a specific omics data from patients or cell lines. We get a robust digital twin that can simulate the effect of different drugs and predict synergistic drug responses using the DrugLogics pipeline. So my project is about optimizing the process of data integration and the analysis of the outputs. And my aim is to integrate all these steps into a user-friendly platform. Then, how does the DrugLogics pipeline work? It gets input as biological data, and it works by taking this as a list of drugs that can have their respective targets. Then the drug combinations to be tested. Then the omics data represented in scale values 0 to 1 as initial states of the entities defined in the network. That could be a zip file of the network topology. And then it executes in a Java launcher 2 packages, the Gitsby and the DrugMe packages. And the first generates a library of Boolean models that can comply with the omics data. And then the second one performs the combinatorial drug responses and is able to identify synergistic combinations. And from this, we can get a biological testable hypothesis. What I'm taking as a case of study is the JAX drug screen study that was published in 2022. They have tested 125 cancer cell lines from 3 types of cancer, and they have tested more than 2,000 drug combinations. Now, they have used an anchor approach and where they have tested different concentrations for the drug combinations. And they look at these responses and determine what is a synergy and what is not a synergy in these cases. Then I take this data and use the flowback AGS model that is a Boolean model based on gastric cancer cell line, and it has, even though it's only for made for gastric cancer, it has the common pathways that are affected by many cancer types. So I take these two, and then I work by translating this biological data into pipeline files through Python scripts. So I map the names of the drug names to IDs using an inside tool called Drug Profiler inside from the research groups. Then I make a list of drugs and combinations with the respective targets, using the Drug Profiler tool. And then I map the node names to the HGNC symbols so they can be connected to the omics data. And then I scale the values. We scale the values using the decouple R2. Then we run, when these files are ready, I run the pipeline and the results are compared to the lab results. And in this way, we can evaluate the predictive performance of the models and the pipeline in general. So as preliminary results, right now I am working in the analysis scripts to do this more automatic. And I compare the pipeline results of the observed JAX screen to the pipeline synergies through ROC curves and precision recall curves. And as for today, they don't look very nice, but as I say, I'm still working on it. And finally, my future steps are to evaluate different models and also evaluate different drug screens datasets, and finally integrate everything in a web interface. And that's all for today. Thank you for your attention, and I'll be happy to answer questions at the end of the webinar. I think I went too fast.
Thank you very much, Vivian. So now we'll move on to Marco.
So I'm Marco Fariñas, a PhD candidate in systems medicine at Kais Faletis lab at the Norwegian University of Science and Technology. And today I'm going to present about my PhD project, which is about computational modeling of high-grade serous ovarian cancer. And specifically, I will focus in this presentation in how I'm generating training data from multi-omics datasets and how I'm trying to discretize it and integrate it into the model. A little bit on high-grade serous ovarian cancer. Ovarian cancer. Ovarian cancer is the most lethal gynecological cancer. It stands for the eighth leading cancer-related death among women. The most common type of ovarian cancer is epithelial origin ovarian cancer, and high-grade serous ovarian cancer is the most common type of epithelial ovarian cancer. High-grade serous ovarian cancer is typically late diagnosed, being the 80% of the patients diagnosed around the stages 3 and 4. High-grade serous ovarian cancer related deaths constitute around the 70% of ovarian cancer related deaths. And also the 5-year survival rates are pretty low, 41% in the case of those patients diagnosed at the stage 3, and 20% of for the patients that are diagnosed at the stage 4. Regarding the mutational profile in high-grade serous ovarian cancer, what is more important here is the chromosomal instability, and also we have some genes that can exhibit somatic mutations. The genes that are affected either by those kinds of mutations are related to stress responses, cell cycle control, MAPK signaling, the PI3K-AKT pathway, beta-catenin pathway, cell growth, and the DNA repair. However, in a tumor, we don't really find only tumor cells. We have accessory cells from the immune system and the stroma that are constantly segregating and expressing a complex interplay of mediators that are recognized by the different cell types, including tumor cells. So the tumor microenvironment really constitutes a variable that can define the aggressiveness and ultimately the phenotype of ovarian cancer. In my project, I'm using Boolean models, and these models are made up of a network that is integrated by nodes or components that are typically genes, transcription factors, signal transducers, and interactions among them that are known as edges and can be either positive or negative. Also, we call these Boolean models because the activities of these nodes are defined by either 0 when the node is inactive or 1 when the node is active. And in order to define the potential activity of a node, we use logical rules that make use of logical operators and, or, and not. So if we take a look to node G in the network, we can see that it's positively regulated by the node C and negatively regulated by the node I. So in this case, G is going to be active when C is active and at the same time I is inactive. So basically, by defining an initial state and the logical rules of the model, we can perform simulations of the model dynamics and generate an output that can be eventually linked to a biological phenotype. And the workflow I'm following in this project is to refine a high-grade serous ovarian cancer network, to annotate nodes and edges using available literature, to generate training data, to generate logical rules, analyze dynamics, find targets, and eventually to study cancer stroma crosstalk in a bigger tissue model that will use this Boolean model as an input, together with some quantitative parameters, such as stiffness or cell proliferation rates, for example. So since I'm focusing on cancer stroma crosstalk, each of these steps has been developed from a tissue perspective. And so far, I've accomplished the first 2 steps, and I'm currently working on how to generate the training data. So this slide includes a caption of the model that looks like this, and it's just to illustrate that it's made up of different modules or pathways that are relevant for cancer and ovarian cancer and high-grade serous ovarian cancer itself. In addition, each of the nodes and the edges have interactions, so the network serves as a knowledge database itself. And now I'm working in generating the training data. And what is the training data? It's a Boolean vector that defines node states that serve as initial conditions and will guide logical rule assembly. This is basically a vector of zeros and ones that are inferred from experimental data. And typically the experimental data that we use is multi-omics data. And in this project, I'm focusing on two types of omics data: mutation data and transcription factor activities that are derived by transcriptomics and single-cell transcriptomics. This is because they are easy to Booleanize. Because, for example, in mutation data, we have mutations that lead to a loss of function of a protein or a gain of function of a protein. And this is easy to Booleanize. In the first case, if a protein loses its function, it's gonna have a 0 associated to it. And if the protein has a gain of function, it will have a 1 associated to it. And then the transcription of course, these mutations must have clinical relevance. And regarding transcription factor activities, this is based on the premise that if we have, for example, a transcription factor A that encodes the expression of 5 genes, and upon specific conditions, these genes are upregulated, we can infer that the activity of this transcription factor is increased. On the other hand, if we have a transcription factor B that encodes also for the transcription of 5 genes, and they appear to be downregulated in this context, we can infer that the activity of the transcription factor B is decreased in this case. So the workflow I'm following to infer this transcription factor activities will end actually with the use of decouple R. So yeah, in more detail, the workflow is that I'm using data, public available data extracted from two databases, GTEx and TCGA. The first one is for collecting samples derived from healthy ovarian or fallopian tube tissues, and the second one for samples derived from cancer patients. It's important to state that this cohort is free of bioinformatics batch effects. Since we are manipulating tissue samples, these samples are heterogenic and they have a mixture of cell types. In order to study the abundances of different compartments, I perform bulk RNA sequencing deconvolution using the following tools. I use 3 tools in the case of healthy sample derives, healthy patients derived samples, and 4 tools. This is because estimate, the last tool was developed for specifically for cancer. So from the deconvolution, I am generating 3 groups based on abundances of epithelial and tumor compartments for healthy derived samples and cancer derived samples, respectively. And then I will perform for each group a differential gene expression analysis of cancer samples versus healthy samples. And I will study this differential expression gene list using a signature matrix extracted from single-cell RNA sequencing to infer which genes are differentially expressed but originated from cancer cells. And then the last step is to estimate the transcription factor activities using this differential gene expression dataset filtered out by those genes that are differentially expressed by cancer cells, and I will input it to decouple R in order to do so. So these PCAs are shown the results after the convolution. Red dots indicate those samples with high abundances in the left plot, of healthy samples that are enriched for the epithelial compartment. And in the case of the blue dots, represents those samples that are not enriched for epithelial compartment. And in the right plot is the same, but for cancer. Of course, in cancer, we have more variability in both of the groups. And I decided to integrate the results of these tools instead of using one tool alone, because when performing the convolution using individual tools or combining few tools, we were indeed able to see tendencies. However, the filtering and grouping was less clear in this case. So now I'm exploring the differentially expressed genes, and I'm using also a single-cell RNA sequencing data to extract these differentially expressed genes profiles that might come from cancer cells that I will input for the transcription factor activity inferred tool. And next steps will involve to once I have these transcription factor activities, together with the mutation data, generate training data that will allow me to generate models and logical rules of these models, and then analyze its dynamics. So this would be all from my side, and I'm looking forward to hear your questions in the end of the presentation.
Thank you very much, Marco. So now we'll move on to Malvina.
Okay, so hello again, everyone. I am Malvina Marco. I'm a postdoc in Cancer Research Center of Toulouse, working in Pancaldi's lab. And here in this talk, I'm going to speak about the approach that we have been following to build a multi-scale dynamical modeling following a data-driven approach. So in our, okay, this is not the, perhaps the audience to explain what are the gene regulatory networks and why is it so important to study them, but basically they model or they represent the regulatory functions between genes that regulates the target ones. And they are composed on two different parts, let's say, or they have the two different components. One is the network structure, indicating who regulates what or which gene regulates what, and the function, the function part, which explains how each gene, each source gene regulates the target genes. So why is it so important to study the gene regulatory network? As Marco noticed, as Marco mentioned, is the fact relies on the fact that once we have a gene regulatory network, then we can use it as a backbone to build dynamical models that can further be used to simulate different environment, either intracellular or extracellular or drug actions. So, but on the other hand, the question goes, why is it so challenging to define gene regulatory networks? Well, to build them, there are two main approaches to follow, like two main pillars. Either you follow a literature-based approach, like searching from published evidence, like the functions and the structure of the gene regulatory network, or you can follow a data-driven approach, like using different types of omics to infer and to build these regulatory networks. But it's not only computationally, like which approach to take. The gene regulatory networks represent different levels of control, starting from the chromatin state and then going to the transcription factors and the signaling networks that are different levels of regulation. And on the other hand, they are very context specific. So they differ from one cell type to the other one, from one individual to the other one, from one tissue to the other one, and one disease to the other one. So let's say that a global gene regulatory network for a certain cell type perhaps do not exist, but we do what we can. So in our group, we studied the tumor microenvironment. So we want to know more about this ecology of interactions between tumor cells and immune cells. As we know, of course, the TME composition it is tightly linked to tumor progression and resistance to therapy. And we know, again, that there is a lot of crosstalk, not only between cancer cells and immune cells, like the immune cell types that are present in the TME, but also a lot of interactions between immune cell types and their specific phenotypes. And in our lab, we had a particular interest in macrophages. So macrophages are one of the most abundant immune cell types in the tumor microenvironment. They derive from the differentiation of monocytes, and they are known to have a very plastic. They are very plastic cells. So depending on the signals that they receive from the extracellular environment, they can be polarized to different phenotypes, starting from a pro-inflammatory M1 phenotype and going to an anti-inflammatory or M2 phenotype. And particularly when they are in contact with cancer cells, they can display this phenotype called TAM, which stands for tumor-associated macrophages. And they have a pro-tumoral phenotype, and they can protect the cancer cells from going to apoptosis. So on the other hand, what do the cancer cells do? Like immune cell types are very plastic, but on the other hand, cancer cells as well can display a lot of cell states, depending on the conditions of the TME that they are in. Starting from, they can go to apoptosis if they do not have the right conditions to proliferate, or they can activate proliferation pathways, or they can undergo to metabolic shifts, etc. So so we were interested in studying how the interaction between the cancer cells and macrophages, but in general other immune cell types, can define the behavior of the cancer cells. And I didn't mention, but the cancer type that we are interested in our lab, or we are studying in our lab, is chronic lymphocytic leukemia. It is a blood disease characterized by malignant B cells. They proliferate into the lymph nodes and then can circulate into the blood. And again, to study how the presence of macrophages or other immune cell type define the behavior of cancer cells, we have performed different in vitro experiments. In the first condition, we have extracted only the CLL cells, the chronic lymphocytic leukemia cells from patients, and let them culture alone. Again, in another condition, we have mixed cancer cells with macrophages, so we could see how the protumoral phenotype of cancer cells could affect the viability of cancer cells. And in the third condition, we have performed autologous culture from PBMC CLL patients. And from that, we have performed bulk RNA-seq in several time points, selected from discussions with biologists who knew the system a little bit more. So with that, we wanted to follow a system biology approach, like to understand how gene regulation could explain the behavior and the phenotypic appearance of cancer cells, like the phenotypes that they display along the time course, or the cell states that they undergo. So there were a lot of steps that we have been taken for the scope, with the goal, the main two goals is to infer the gene regulatory network of cancer cells in a completely data-driven approach, with the aim to apply on top of that a Boolean models that can then be used for simulations of different perturbations or different conditions. So we follow this, we use these three conditions as a way to study a kind of which trajectory the system would go in the presence of not of immune cell types in the plate, like in interacting with cancer cells, as in starting from a certain initial condition, like all the cell types would be more or less having the same being in the same condition, but then the presence or not of immune cell types would lead the system or the cancer cells to go into one of the trajectories. And we could see actually from the three conditions a clear separation between the three. So there were a lot of questions that we were posing when starting this project, whether to perform a context-specific GRN, like to infer a GRN specific, context-specific for each of the cultures, or to build a general GRN for the cancer cells, and then to follow a data-driven or a mixed approach, like to mix it a little bit with what is already known, and then to undergo, like to follow a study on analysis on temporal gene regulatory network or dynamical modeling. So here I'm going to present the results that we have obtained so far in this project. Before that, previous work that we have been done in our lab, we have already done it. We have already built a gene regulatory network and the Boolean model of macrophages, consisting on a GRN with different extracellular signals, a lot of genes like intracellular ones that whose expression could give us the appearance of different phenotypes of macrophages M1, M2, and could observe a certain or a separate phenotype of macrophages that were specific in the chronic lymphocytic leukemia, which is the analog of tumor-associated macrophages. So for the cancer cells, having the time series of RNA-seq, we followed the data-driven approach. Basically, there are several algorithms that we can use or one can use for inferring GRN from time series of RNA-seq. And we have performed a kind of review of available methods or published methods for this scope. The method that we choose to run with is called DinGene3. In the later slide, I'm going to give more details about how the method performs. But what is the output of it? It is a directed GRN and weighted one. On top of that, we have performed some additional analysis like gene set enrichment analysis and structural analysis as well. So briefly, how the algorithm works for inferring the GRN from the time series, like having the time series and, if available, also the steady-state data set in different conditions. The expression of a certain gene in one of the time points, it is modeled as an ODE function of the genes at the previous time point minus a certain degradation rate. And this ODE function, then I learned through random forest algorithm, whereas the degradation rate can be chosen depending. I mean, it's a choice to choose between either it can be gene specific or it can be a general one that we can set beforehand. In our case, we estimated the degradation rate for each of the genes and then selected the mode of these alphas for running the algorithm as a general degradation rate. So the result that we obtained, of course, we have done it, this network inference for each of the conditions separately, but then aiming to build a general gene regulatory network. This is on the left side. It is the merged regulatory networks from the three conditions, where here the colors indicate the genes that were specific for each of the conditions, and the red ones here represent the ones that were in common between the three conditions. So we could see that, of course, there are some that were in common between the three, but also there are a lot of TFs that were condition specific. So that set of genes, or those set of genes, could give us more information, like how the cancer cells are not behaving, but how they are interacting with the environment surrounding them. Furthermore, so this work was performed mostly by Hugo Schenel, now postdoc PhD candidate in our lab. He also performed some gene set enrichment analysis on based on the genes of this network. So we could see a lot of pathways enriched, indicating or like pathways related to cell communication, production of cytokines, but there we had also a lot of them following indicating proliferation, activation of proliferation pathways, and a lot of metabolic shifts of cancer cells. Perhaps this is a kind of related to the nutritional environment or the conditions of the cancer cells during the time course. So actually this part we used it afterwards for inferring the Boolean models, like to look for the phenotypes of the cancer cells to look at. So as a next step, we wanted to go from this static network, just assigned one, to a directed one. And for this reason, we used two sources of information. First, we used College Tree database, a newly published database for, it is a database for signed TF target interactions. So from there, we used the big network of TF and target genes, which is a signed network, and selected only the ones or the interactions that were in our inferred GRN. And for the interactions that were not present in collect tree, then we use the TF expression correlation. So the scope was to go from just a signed network to a signed, the idea was to go from a directed network to a signed network. And this was the signed network for the CLL, for CLL cells. It is the same network as I showed before, but just here we have the how also, like the information of how the genes interact, like whether it is a positive or a negative interaction. And the scope, it is that on top of this network, we can then apply, or we can build we can apply dynamical models. And the dynamical models that we were interested in is Boolean models, like Marco made an introduction before. Again, for the scope, like to build these dynamical models, again, we wanted to follow a data-driven approach again. For this scope, we're interested in using Bonesis, which basically takes a prior network, like initial GRN that we have already inferred, and it takes the initial conditions, like where the system starts from, and having some prior knowledge about the states that we want to look at, the steady state that we want to look at, or we want to be interested in. And as an output, it infers, or it builds a ensemble of Boolean models that explains the GRN and the transcriptomics data that we put as conditions. So this is still work in progress. Work in progress, and some ideas that we are having in applying is to look for specific phenotypes of cancer cells, mainly related to apoptosis or proliferative, or these metabolic shifts that we have been seeing also from differential gene expression and TC-seq analysis. So what is the global aim, or what is the global scope of this project? It is to build these multi-scale dynamical models. Like the thing that I didn't mention about the transcriptomics dataset that we have been collecting from our experiments is that so far we have been working on bulk RNA-seq in time series, but we have also performed site-seq time series for the same conditions, with the idea to follow this workflow from a data-driven network inference to Boolean modeling for each of the cell types that we have in our network, and then adding on top of that the metabolic part of the cells, and then going step by step, or scale, like in an upper scale, toward cellular modeling when we build dynamical models of cell interactions that we observe, or we have in the in vitro system. With that, actually, I am very happy to announce that this project, with using the site-seq experiments or datasets for this data-driven multi-scale modeling, is now funded by NTNU in collaboration with Loïc Paulevet and Laurence Calson. And I am looking forward to working and to start this project, but and I'm happy also to take questions about the current results that we have been that we have obtained so far. And thank you very much.
Thank you very much, Malvina. So we'll take questions, or let me just share my screen for a moment, because if people want to ask questions for one of the specific presentations, you can just write the name of the speaker, or write 1, 2, 3. So you can see it here. You have Viviam, Marco, or Malvina, or 1, 2, 3. But if you ask more general questions, that's also okay. I will see who of them wants to answer them. So you can go to the Q&A function of Zoom and post your questions there. While you do that, I'm gonna start, and I'll start with a question for Vivian. Vivian, you said that you want to make a user-friendly interface for your DrugLogics pipeline. Yeah. So how do you envision that work? How will you make sure that it's user friendly? Will you ask your users at some point? Will you have some user testing or something like that?
Yes, it's one of the points during the project to have use cases and have users trying the platform and see how it goes. And I think the aim comes because I don't have that programming computational background, more in biology. And I feel like this is something that needs to be more available for the general biologist and for the general people that work in the lab, so they are not that scared of using these tools. And that is my aim. And yes, we will have some use cases and users trying to do this. Yeah.
Okay, thank you. And then I can continue with Marco. Oh, there's a question there, so let's go for that. There's a question for Marco and one for Malvina. Marco, the percentage of late diagnosed ovarian cancer is surprisingly high. Is this because the onset of the disease develops rapidly, the biomarker knowledge remains limited at earlier stages, or something else?
Thank you for your question, Aline. So high-grade serous ovarian cancer is a bit special. So this type of ovarian cancer is aggressive itself. So its cells really migrate quickly, and also the detection methods nowadays, state of the art, don't really allow early diagnosis. That's in combination of this and the aggressiveness of the disease itself. Leads to late diagnosis and overall low 5-year survival rates. Also, the ovarian cancer, the cancer establishes mainly in the ovary, also in the omentum and peritoneum. However, now it's known that these malignancies actually originate from the fallopian tube and then they migrate toward the ovary, which is the closest location. So actually primary tumors, or what are defined as primary tumors, are already metastatic.
Okay, thank you, Marco. So Malvina, what kind of reasoning can be done in the Boolean network to be able to perform model reductions? And do you focus on the network topology or model dynamics?
So network topology and model dynamics, I would say, okay, I will give my honest opinion on that. Like, I still find a gap between the two. Like, how do you use network topology to understand more about dynamics? And can we infer, for instance, what is going to be the dynamics of the network based on network structure? We have performed network analysis on the networks that we have performed. We have performed structure analysis on the networks that we have performed, but so far these two analyses can give only. There are still separate ones. As I said, there is a gap between the two. So if I were to perform model reduction, I would follow the classical way, which perhaps from the biological point of view should be revised, meaning reducing as much as possible the intermediate nodes that just transfer the information. But that sometimes biologically it is risky because it can like specific nodes, although they are just intermediate ones, can have a lot of important information. Then and the reduction should be revised from experts. So so far, I mean, like, we have not risked to perform network reduction, but that was the way that I would go.
Okay, thank you, Malvina. So Viviam, there's also a question for you. How would the user of your interface be? Who would the user of your interface be? You said biologists, why not clinicians?
Yes, definitely clinicians will be one of the users. I say biologists because this is the first thing I think about. But yes, this at the end will be a tool for the clinic and is trying to predict better treatments for the patients. But then in that case, for the clinicians, they need to have a bit of training on what the tool does and how to interpret Boolean networks, or maybe try to train them or educate them in Boolean models and omics data integration. I mean, it needs to be user-friendly, but you also need to have some knowledge to use it.
Okay, thank you, Viviam. And talking about the tool, will people be able to contribute to the data that it's in there? I mean, do you envision someone doing the same as you're doing now with all the data and putting it together and adding it to the pipeline?
Yes, I have a lot of ambitions with this one. I will not be able to do everything during my PhD, but I think I would really like to see this tool with the work of Marco and the work of Malvina, for example. How can we integrate, like, after Malvina developed the model and everything that is, like, she's confident on the network, can we plug it into the pipeline, and can we see what is on drug prediction? Can we assess, can we analyze it with that? So definitely I want like more plugins into the tool. My project is to try to do the basic things, like plug in or integrate the basic things, but I definitely aim for much more.
Okay, thank you. Okay, so I can ask you some more questions. Marco, a question for you. And this is a question that I always think about when I hear about models and all that. So you are doing all this analysis. You want to generate these models. And once you have your models, do you think about maybe doing some experiments to validate some of the predictions, or to try to find something else from starting with the predictions that your models make?
Exactly. Yes. So the process of modeling, predicting, and validating is cyclic. So at the group, we also have an experimental core. And at this stage, what I did is to design a network based on a network that was already existing. And now I'm inferring these activities to calibrate them and generate models based on experimental data. However, in order to trust that the outcome, the model, the chosen model is predictive, I will definitely need to validate in the lab. So we will perform directed experiments in order to test whether the model is predictive or not. And if it's predictive, then we can move on. Otherwise, we need to go back and do some refinements.
Okay, thank you.
Can I ask a question?
Yes.
Another question, Marco. You were in one of the steps of the workflow that you want to follow, you wanted to make a differential gene expression between healthy donors and patients and cancer patients in TFs, right? To select the most differentially expressed TFs. So I was wondering whether you were considering also to do it on TF activity more than TF expression.
Yes,
because
yes.
the TF expression per se perhaps might not be so informative as
Yes, exactly.
the TF activity.
Yes, I will use the output of differential gene expression analysis as an input to the couple. Then I will use actually collectry in order to infer the transcription activities, and those will be the ones and zeros that I will be inputting to the model as training data. So yeah, yes, I decided to go to transcription factor activities indeed.
And how are you think that you're going to deal with patient variability and initial condition?
Yes, so what I'm making is a big, big list of comparisons. As I presented, I'm making, so I'm trying to focus also in quality control. For that, I was focusing on sample composition regarding cell types. For that, I did the convolution, and then I'm making comparisons between the groups. Cancer versus healthy. Then I'm doing comparisons also intra-groups, meaning like low, high purity differential gene expression to check the variability of the same groups. And then I will do also comparisons using a big control group and each of the patients individually. Mm-hmm. And then by combining the outputs of the different cross comparisons, I will be more confident, let's say, on the outputs that I'm generating and the inputs that I'm using in further steps. Yes, a lot of quality control, I would say, and semi-automatic processes.
And if I can interrupt or
Yes.
go into what Marco will make, or the results of Marco, is something that I will take in the pipeline, Malvina. So depending on how he figures this problem out, I can also move forward, because my aim is to integrate the omics. But it's the same problem, like what is it that we are going to integrate? Are we going to integrate gene expression or transcription factor activity, phosphoproteomics or metabolomics? What is it that we are going to integrate? And if so, then the next challenge is how do we scale those values that are continuous values in real life to something that the Boolean model can interpret in zeros and one. So I think we have this challenge. We will try to do the best, but still I feel like we are so far. I mean, there's too much that we cannot control or we cannot predict right now. We need to make the first steps. But your point is quite right. We don't know yet how to get this going.
Okay, so if I can ask one more thing to Malvina. Malvina, you have the cells, like the cancer cells and all this microenvironment, and you focus especially on the immune system, macrophages, and so on. But you're trying to integrate everything in the model. But at the end, how do you envision, if at the end this has to end up in some treatment or so for cancer patients? Do you think it's going to be more towards the immune cells, towards the cancer cells, or it's going to try to go, to how to say, to attack in a way all the cells that are there involved in the cancer?
Yeah, so thanks for the question. It is I would say, like, okay, it is still too fast to come to this conclusion, but I would say both ways. There are some, like, we can simulate whether the model can reproduce some modes of action of known drugs that are used for current lymphocytic leukemia. But also some collaborators in our institutes are targeting mostly immune cells, as particularly macrophages and this protumoral phenotype of macrophages, TAMs, by how to say, like, developing antibodies that can detect them and target them by killing, like to kill this phenotype of macrophages, because it is not that they protect the cancer cells directly, but they disrupt also the function of other immune cell types. So other immune cell types are less effective in fighting cancer. So by targeting them, then perhaps we can optimize a little bit the immune system to be more effective against cancer. But again, like, I would not be preferential to one, like whether to target the immune system or cancer cells. My scope is to build these models that can reproduce what is already known, so that we can use it for further in silico experiments on both sides, as I said. So, but this is still, like, the global aim, but we are working on that.
Yeah. No, but it's like we have to work little by little in this. I think
true.
research is not done in one day. So but it's I think it looks really good on what you have presented today and how we are using these computational models in the research for cancer. So we have still a couple more minutes if anyone wants to ask a question, or if one of you wants to ask a question to each other. But I'm gonna just show you a couple more slides. So the PerMedCoE webinar series continues running, and we are going to have our next webinar on the 7th of November, where Marco Ruscone from Institute Curie will present multi-scale model of the different modes of invasion, a PhysiBoss application. So you can already sign up for this webinar on the PerMedCoE website, and we can then meet you again next month for this webinar. And then I would also like to show you this course that we are organizing at MBLBI in March next year. It's a virtual course from Monday to Friday, a whole week, on mathematics of life, modeling molecular mechanisms. And it's especially aimed at people who are starting with modeling. So the idea is that it's an introductory course, but you will gain some experience by working on a project on modeling. So if any of this is of interest for you, you can go to the ABI website and take a look and register for it. So I see there are no more questions. So thank you very much, everybody, and see you soon. Bye.
Thank you all. Bye.
Thank you. Bye-bye.
Thank you. Bye.